where we're going to take a stroll back the memory lane. And now we have for 2 days been having these fantastic talks and seeing great posters and the state-of-art research with our favorite fungi. And I think you will all agree with me that the quality of research, the development of the research in our field has really taken off the last 10 years. And I think it's important to keep in mind that that has been possible because of some pioneering discoveries that were done, some pioneering development of methods that were done, understanding the infection biology of our organisms, understanding the contribution of sexual recombination. So some decades ago, many of the pathogens we were working with were just considered to be asexual, and now we know that sexual reproduction plays an immense role. It's also about establishing experiments, establishing protocols for working with these organisms in the lab. And one thing I want to mention, because many of us have been profiting from the ECO323 genome, and I'm sorry for being here, Simon Victoria Trinity bias. I think today it's one of the best eukaryotic assembled genomes, and that's because of a genetic map that was done with some of the first process of Simon Victoria Trinity. So without that work, we would not have that genome resource. And then there is another thing. So now some of the talks here we've heard about collections that are being done now, and then also old collections that have been reduced. And our understanding of the population biology of our favorite pathogens also only rely on these collections in the field, which I really want to encourage all young people to continue to collect the pathogens, because this is immensely important material. So Gert, Marc-Henri, Richard, Steve, and Bruce have really been pioneering the research that we are doing, and so that's why we wanted to have this special session here. There is one important person who is not here today, and that is because she takes it very serious about retiring. I should jump here to this picture here. So we invited Lisa. So of course there's a strong woman behind everything here also, and we invited Lisa and Ms. to come. So she's been part of the community and part of driving in particularly discoveries or developments in applied context forward. And I want to mention Lisa not only because she cannot be here today, but also because Lisa, she's retiring now with a fantastic achievement. So for the last few years, she has been using some of the knowledge also on the population biology of the leaf pathogens to bring together breeders, stakeholders, and farmers, and to develop a program for mixed cultivars. And today in Denmark, between 40 and 50% of all wheat that is produced is based on mixed cultivars. So that's a lot. I know that France is also leading in that respect. And this has significantly reduced disease pressures. So I think that's at the end of the day is one of the achievements of our research. So I think it's important here to mention these things, unfortunately, cannot be here. And with that, so we are just going to, so maybe also for the young people in the audience, this is the first time that you participate in an ISCLB meeting, and you get also probably, just like me, I always get confounded with the order of letters. What is this here?
Before these guys get started, we just want to show a little bit of the history of this meeting. And actually, I didn't realize it went back this far, but these guys were not present at the 1976 meeting. So this was started by a group of mostly U.S. scientists in 1976, and it's had several names over the years. Just the last time it turned into ISCLB, which maintained this time. The first meeting that I went to was the Tunisia meeting. And I met all of these guys, except for Marc-Henri, he wasn't there. I met him then again at the Tunisia meeting, or the ISCLB meetings. But I have to say, these guys have been mentors. They've been good examples as scientists. And I counted up in my in my CV, and I think I have 38 publications with people that I've met at this meeting for the first time. So this has been a really good meeting for me. So as young scientists, look around at the people that you'll work with for the rest of your career. It's been a good thing for me. So I just wanted to show a few pictures here from, this is the first meeting that I went to. The things that I remember about this meeting are not in this picture. I remember these two over here on the cell phone trying to get funding for the first Septoria or Septoria genome sequence. While they're at that meeting, I think there was a deadline for NSF or something to that. But you can see how much younger they look here. Bruce is working on I don't know if that's an Apple IIe there. But definitely, I do think I brought a slide deck to this meeting to give my talk, and I talked about the first work that Justin and you and I had done on TOX1 SN1 interaction. So this is the group at the place that we visited. I think it was called Goodwin. Yeah. We visited. That was really cool. And these are you can see these are 4 of the 5 guys that are here. And then this was the ASCLB meetings, and this is where we started to get, so Bruce introduced the bell there. And you can see some of the newer faces that are starting to come in, and these have become the people that are giving the lead talks here. So it's been fun to watch the people develop at this meeting. So this was one of the hikes that we took from here in Stone. So with that, we're going to start with Gert and give them an opportunity to talk about some of the stuff that they've done over the years, give a little historical perspective, and try to keep them on time, but I'm not sure it's possible, but we'll do our best. Here's here.
Do I need a mic? Can you hear me in the back?
Yeah.
Yeah? Sure. Okay, well, first of all, thank you very much for inviting the old block, so to say. So it's a pleasure to be at this meeting again. I just came in last night, so I wasn't here for the entire meeting, but it's a pleasure to be here today and to listen to your talks. And I'm thankful for kicking this off. So I'm not giving you any presentation on deep science, but just a kind of an overview of where we're coming from. I think a few of the questions that were asked is, okay, just tell the audience a little bit, who are you, what are the remaining questions, and what you're currently doing, or what are you planning to do. So I'll give you a snapshot of that. Good, next slide. So folks, for those of you who do not know what IPO means, so IPO is the Dutch abbreviation for the Research Institute for Plant Protection. That is this building. So I started here an internship in 1980, and every master's student I meet and tell, look, your internship can really change your life. And it did change my life, because here I met a person who told me that he worked his entire life on spikers, and I just couldn't imagine that you can work your entire life on single fungus. I didn't come from an agricultural background, plant pathology was totally new, but I loved it. So I have my office here, and I started here working. This parking lot I also never forget, because I crashed the car of my sister on the parking lot. Just driving through 2 fire cars and boom, collision. So this is in Wageningen. So IPO then, don't forget it, Research Institute for Plant Protection. And when I think about it, now remember why did I come up with this title on IPO 323? Because I read a paper by Marc-Henri and French colleagues. And what you probably should know is, when I started working with French breeders on Zymoseptoria, guys, there wasn't a single French person working on Zymoseptoria. And so now that has totally changed, this is great, right? So there are new communities, new groups, but I was kind of, building the path for the French guys to start working on Zymo. And then they wrote in the paper, yes, IPO 323 is a European island. I said, forget it. It's not a European island, it's a Dutch island. So there we go. But some of you think I started designing Sartoria. That's not true. As I said, I started with Cypress. So this was in 1980 or 1982. And then I studied pen reading in 1994. And I started working on my designer work actually in the late 80s. So, and I tried to summarize it. I think everything we did was kind of revolving around the question of specificity in sex. That was the whole story that was really intriguing in those days, because in design Sartoria we decided we just didn't believe there were gene-for-gene interactions. Right? There were even suggestions that it is a soluble pathogen. And it wasn't the least professor who mentioned it, for instance, that was part of it. So that was the father of the overall resistance in cereal crops. So they were all. Anyway, what I started doing is, what you see on the left side in that table is just phenotype. Multiple varieties, multiple isolates. This table was, I believe, 25 varieties, 50 isolates. That was simply an awful lot of work, but I saw a lot of phenotyping coming right this afternoon as well. We all know that phenotyping is laborious, but it's also nice because you're on your own. You see all those interactions, and of course, during observations, you keep on thinking, right? And but there was a lot of hesitation to accept that specificity occurred in this sample system. And then I decided, you know what, I just can't prove it all the time by just doing more of those phenotyping. It's just more. That's not convincing anybody. So let's make a selection of isolates and go to the field. So this is a field experiment where I tested individual plots, individual varieties with individual isolates. And Alice Turte last year, very nice. There you are. Yeah, so she showed this picture as well and said, okay, you know, two guys in a wheat plot. I said, okay, probably I'll give you a little bit more background. Because this here is James Brown. And James Brown was probably the most prominent critical of specificity in Sartoria and weeds. He said, well, come down, look at this field experiment. So he came over, I think it was 5 minutes in the field, and he said, you're right. So that was a total conversion. So he got hooked on Sartoria as well. We started to collaborate, and then we started to map first resistance genes, and it started, I think, with FTB6. So that was the field experiment. So this is James, and this is me, just to show him the individual isolates and manipulations in the field. This was in Arnhem. Okay, then we come to IPO 323. So this is an old card box piece of paper. I have it actually framed in my living room. What it says, so and remember it's a Dutch islet. It was sampled in 1981 on the 6th of May by Richard Damen in the field of farmer Klaassen from the cultivar Arminda. And it was isolated by one of our technicians. And how did it come across this isolate? As I said, I selected isolates to work in the field. But of course I had to take Dutch isolates. I was reluctant to take an isolate from Uruguay and put it in the field in the Netherlands. So I just more or less randomly chose a couple of isolates. I had tested them already in the seedling stage. And there, IPO 323 was really an outstanding example of differential interactions because it was very clear. It was either super pathogenic or avial on maybe 20 varieties or so. So okay, let's see whether I can reproduce that in the field. So that worked out. So just for you to remember, this is the reference IPO 323, a Dutch field strain. And as I said already earlier on, you know, you could continue with phenotyping, but that eventually will not convince anybody on specificity. So then Ricardo Madariaga told me how to discharge ascospores from wheat stubbles, and I started just very pragmatically to see whether I can make a process protocol. Well, that all failed, and eventually I simply thought, okay, if I have 8 ascospores that are pretty close to each other on an agar plate, it's probably from the same ascus. I just take one of those isolates, one of those spores, and combine it with these 7 others. So then I have a 50% chance. If there is sex, I should have a project. And that worked out very well. So immediately an ascospore slammed to the agar. You know, you just immerse it 15 minutes, and boom, you have one in the agar. So this totally opened a new window for me, and I think also actually for the community. And so I still remember that moment when I sat behind my microscope and saw these ascospores and said, wow, and this is really something. Now I see something that never somebody hadn't seen before. So that was very stimulating as well. So and from there on, we started to continue building genetics in Sartorius redesign. And of course, it started by, looking at the inheritance of averins, how that worked out, and eventually many others. And of course, eventually, as Eva said, netting. And we used also dark markers, so those dark markers could be sequenced. And of course, that led eventually to the fungal genome sequence, IPO323. So as Tim said, I was, I think, the only person with a cell phone in Tunisia, so we were calling like crazy to see whether we could submit and needed time for that proposal. That didn't work out, but eventually JGI embraced it, and this was actually the basis of their Dophilomyces community sequencing program. So many Dophilomyces have been sequenced since then. So that was really, as you can see, a super proactive effort. 57 co-authors, it was a fantastic experience. And I'm still very thankful that we could do that. And of course, what is also beautiful to see that in just over 10 years, you end up on a paper where you have thousands of fungal genomes. I mean, I think that's also absolutely exceptional. So I still would like to congratulate anybody who was involved there as well, because this really lifted Sartoria to a model system for population genomics. And I think that is truly worth underscoring. It's of course a joint effort, but it's also an achievement. Okay, this is only in a nutshell, so I just wanted to show you, okay, this is the origin of IPO323, and I think maybe due to the genetics, we really could lift it to a reference and have a sequence. And that's also what I heard this afternoon, so I thought, okay, 1,000 fungal genome sequence, maybe there are others that are becoming the reference, but apparently 323 is still the reference. Fantastic. So this is one of our latest papers, and what we found out, and I also heard it back in some of the talks here this afternoon, is that you can use fungicides, and so Anne-Sophie Walker mentioned, we have a lot of fungicides used in Sartoria. I'll give you another example in a minute, on another crop. For 3 applications, in wheat is considered a lot of fungicides. But what we found out in this paper is, no matter what you do, I think that's a nice slogan to remember, no matter what you do, if you control the disease by resistance or by fungicides, you can't stop sex, folks. It just continues. So, that's pretty easy to remember, take home message maybe even for you guys, right? So when you spray fungicides, the fungus will have sexual reproduction. It does it on resistant hosts. And Frederick, you showed even that 2 avirulent islands can have sexual reproduction. So that whole mating black box, there's still a lot to discover. And so a few remaining suggestions. I think there's still a big need for an effective in vitro crossing protocol. That will be fantastic, because then you can use also transform strains. This is not the first time I say it, so apparently I still have to repeat it, right? So it's not yet done. But that's important. We have to better understand sex. We still have lots of remaining questions. And that's of course also for other pathogens, but for sure also for Sartoria. I was totally flabbergasted that STB16Q cracked down so quickly in France, because we discovered that gene, we mapped it, it was resistant to 200 isolates. So not a single strain that was virulent in French isolates we tested. So okay, this gene will last long, particularly because of the hypothesis we put in this paper. Because the fungus can have always sex, so you maintain avirulence genes in populations forever. So you just do not expect to see a boom and bust cycle. To our big surprise, that happened in Cereal. And so that is, I think, I saw, when I came in this morning, I saw a paper on STB16Q, avirulence gene, so I understand a little bit what happened, but it's still remarkable, right? So there's such an effective gene, completely cracked in only 3 years. So, lots of questions. And Sarah also mentioned that there's still this big dichotomy between durum and red wheats. And I'm happy she mentioned that 1996 paper, because we published it already in 1996, and apparently that has still not been solved. We tried it by genetics, we also couldn't solve it. But I think that dichotomy is still really intriguing. So it would be fantastic if a few of you would also focus on those questions. And of course, durum wheat is very important. And when I look back at what we did, I think we also addressed quite a number of nice to knows, and maybe not always focused on the need to know. So sometimes you're distracted, right? And Yeah, at least I was distracted, let's put it this way. Maybe you are not distracted, but I was distracted now. Alright, so that is the Zymo story, very briefly. And then it was eventually by love, you know, we had to pack all the isolates in a van. And I was so happy that Eva was very willing to receive that collection. So over time we built up a huge collection, very different from the collection that Bruce collected. It was from probably many more places, but we had a lot of progenies as well from all crosses that we made, and also the knockout strains of a whole range of genes. So I'm very happy that it's now safe in Peel, and that's a resource that can be used by the community. So good luck with it. Of course, what I'm presenting is not my work, right? So you do this as a team. So I'm happy to show the PhD students who worked through years on this system with me. And on the left, that's my technician Eva Stukenbrock. So she was super active, still is around, and I really have to owe her thanks because she was always my right hand in the things we did. And then, of course, the community building. So Tim already mentioned that a little bit. So I met Tim for the first time in 2003. And he saw me calling, he saw me smoking, he saw me drinking, and then he also saw me praying. He said, huh? That's impossible, I won't forget that you told me that. So no, you know, I need to talk to the guy, how does that work? Anyway, there I show the photographs in the lab of Harold Floor in Fargo. So I was very happy to go there and to be really in the place where Harold Floor did his work and developed the gene-for-gene hypothesis. And then on the right-hand side, Bruce was always super convincing, right? So we were there in the PAC meeting, and then he said, let's go to La Jolla, because I want to show you the green flash. You know, sunset, believe me guys, we go to the beach, and when the sun is touching the sea, so to say, there's a very short moment that's called the green flash. Now, we have been in that beach for, I don't know how long, 2 hours. I still don't know whether he saw the green flash, and very honestly, I still don't know whether he was serious. You know? Maybe it was just a joke. You still have to explain that to me. Maybe that's just not existent. But anyway, we went
Google it, it's real. It's real. Yes.
Oh, good.
Google it.
That was just history. Yeah, I was too sure, I don't know. But anyway, thank you, that is real. But anyway, we went for dinner afterwards, and you see us here on the table, and there I still remember it because that was probably the first time I had crab leg, and crab leg in the Netherlands is maybe something like this or so, right? Well, in La Jolla it was something like this. I mean, it was really huge with a hammer and everything, so I was, you know, something. But you know, with these guys we had not only a lot of fun, but we had a fantastic professional relationship. We were also competing. But you know, it's very good to underscore that you can be competitors and you can still be friends, right? So we really enjoyed that a lot, and yeah, I'm really thankful that I had a chance to meet them. Actually, Steve, I contacted Steve because he was just you were just entering Purdue University. And then he met a colleague of mine in the SIP in Lima and said, oh, I met a guy who starts to work with Septoria. Oh, really? Okay, let's send him an email. Hey, who are you? Can you come over? This and that. So 1 week later he was there. And actually, from all of you, he was the guy who was present at my PhD defense. That was in 1996. And when he left, I gave him the entire hunting population that he had generated, IPO 323 times IPO 95269. Man, that was a treasure. I mean, you would never do that now. He just took it in the plane, back to the States, and we started working. No, really, we started working, and we continued until recently. So, yeah, it has been a fantastic journey, I would say, with these colleagues to build that community. And I'm really honored also to say that this community, and I still feel a little bit part of it, is a fantastic community. It's super friendly, it's collaborative. I will show you another community that is way harder. And so it's something you really can treasure, and it's very good that we are here. Good. And then, yeah, I went bananas. So I want to tell you a little bit about bananas, of course. So I started with bananas while I was still working with Science Tutoria. The reasons are not so important, but I started working on bananas. Major issues, right? So, of course, they are also work with the whole team, right? And I also would like to acknowledge that. So, past PhD students in the top, ongoing PhD students in the middle, and the team at the Laboratory 5 Pathology in the bottom. And of course, in addition to that, the Laboratory 5 Pathology. So, this is also a joint effort. Banana has become really a very important, not a model, but it has been a very important crop. And I'm thankful that we were able to really lift this to an important research group in that role. And that's indeed the other unit I'm talking about. So, talking about fungicide, guys. On the right hand side, this is Winter. Okay? Can you stop this? Because this is not a helicopter that you hear. This is a guy with a mower. I was filming that helicopter and the guy just kept on going. That's the noise that you hear. But anyway, this is, let's see, though, you know, we have leaves every 10 days there is a new leaf. Those leaves are about 1 meter long or something longer even. And as you can see, the disease is absolutely devastating. The only reason why we still eat bananas is that this helicopter on Sophie, 70 times per year, every 5 days. Why? Because this is capitalist bananas. It's monoculture, super susceptible to black seed cobra. And this is what you see, what happens to Fijensis population. So in Costa Rica, 70 times, those populations simply become insensitive to, for instance, in this case, DMIs. Right? You see the same story here in the Philippines. So, you know, coming from wheat, I just don't understand that the whole community accepts that you have to spray 7 times. But of course, it's also us, right? Because we want to eat bananas. Is the time over or what? we're almost there. So next time you eat a banana, don't forget about 1/3 of that banana, the production cost, is sheer fungicides. Okay? So that's also an easy take-home message. The other fungus is under disease is Fusarium wilt of banana. This is absolutely devastating. That's soil-borne. It caused a major epidemic in the 1950s, and actually Cavendish became an important variety because it's not originating from a breeding program. It's a wild clone. There are a few selections with just a wild banana, but super resistant to the strain that caused that first epidemic, and still is. So if you talk about durability of resistance, Cavendish resistant to Fusarium wilt is exceptional, 80 years or so. Now, but this is now what you see. This is what you see when Cavendish is infected by a strain that we call that's a unique genetic lineage. It's a clone by a strain called Propagate 4. Super pathogenic, and that's really threatening global banana production. So it was already identified in the 60s. We saw it for the first time in Jordan in 2013. 10 years later, it spread to 16 new countries. And the vectors is us, the humans, right? This is a soil-borne pathogen that doesn't fly, no ascospores, nothing. If that would happen, we would have a Nature paper, right? So, absolutely asexual, but the spread is devastating, also frightening. So what we did is we also built up collections for both fungi. We are building pensionomes, and Alice and Eva are also involved, right? So we look at diversity of Fusarium populations around the world, and here the search is identify factors that we can use in breeding, to put it shortly. So after this message, you may think, okay, what's going to happen with bananas? Right now, I put those numbers there, 95 and 21. That's just to remind myself that I gave this type of talks in 21 countries, 95 times. You know, just to talk, yeah. I mean, not this type of talk. Of course, about banana research and the need for banana research, and you know that we have to build a community and that we need databases, etc. Now, that is developing, so that's good. But one of those talks was actually, I was requested that was Marc-Henri to give the talk, but then without slides. So after doing that talk, that was just fine. A colleague of mine said, that was kind of like a sermon. I don't know who of you ever attends church, but a good sermon, I think, shows you what's the current situation, how are we doing, but of course a good sermon is also, there is hope, right? So if you talk about the situation in banana, yes, we have to build a community, and we should not forget that we also should be hopeful. So that's why I got involved in starting up a breeding company for banana breeding from the Netherlands. So as you can see, one of the major banana companies is also involved. So what we literally are doing is, despite the fact that we are trying to convince people, said, okay, forget it, we're just going to do it ourselves. So we started a breeding company from the Netherlands to eventually deliver diverse, resistant varieties to the market. And of course, that will cause a major change. So that's a hopeful message. Now I'm finishing. I'm going back to the 2003 meeting in Tunis. I mentioned to you that Tim said, hey, this guy is drinking, he's smoking. I'm not smoking anymore, already for 10 years or longer. Drinking, that was just a glass of beer, right? I mean, it wasn't really drinking, but he's also... Oh yeah, before I forget, what we also started there, Bruce may remember, we started the Marc-Henri Fitness Club. You remember that?
Yes.
Yeah, we started there, but that's something we did not continue. And there's one other anecdote that I should not forget. And that is, I'm sure I'm the only person in this audience that ever had Bruce McDonald on my lap. I'm not joking. That was in Mexico, where we had to put 5 people in a small Volkswagen Beetle taxi that only could fit 3 of us. I said, okay, Bruce, sit on my lap and it's fine. So we arrived there in very direct darkness and Bruce was on my lap. But coming back to the Tunis meetings, I remember we had then invited a speaker who was my promoter, Professor Zano. And when I was there, I thought, you know, I want to go to the site where Augustine lived and worked. So I don't know whether you know who Augustine was, but he's one of the church fathers living in the 4th century. And then my promoter started to joke, said, oh yeah, he wrote a book with Confessions, and he had a problem because he once stole an apple in his life. So he started kind of laughing and said, yeah, that's true, but I can recommend to read that book. You know, Confessions is a classic. The sermons you still can read, they're all translated. But then I thought, yeah, listen, Sarah mentioned one 1996 paper that we ever published, right? But who is reading papers from the 1960s or the 1990s? Not too many people, right? And in one generation, people will just not read the papers anymore. Augustine is still being read. So there's a difference, right? So I think theology, philosophy is very important also for your mindset if you do research. And with that, folks, I wish you God's blessings for all your current and future endeavors. And thank you very much for your attention.
So we are already 20 minutes behind schedule. I picked something your podcast, but
I'm sorry, I thought you were making that.
I think we take the questions in the discussion session. We were already anticipating this would happen.
I do the same, right?
So, Richard, I'm very happy to introduce the next speaker, Mr. Maloro, Richard Oliver.
Well, thank you very much. Thank you very much. Right. So when did you say you got your PhD in? When?
1996.
Oh, right. So my talk is about from Snockbrock to Snocks, but in order to understand that title, you have to go back to my beginning, which was in 1979. So starting out in 1997, I did a PhD in biochemistry. If you think about it, standard sequencing was 1976. The Cohen Boy was 1972. I did a PhD where I worked out the very important thing of how to get a double bond out there, and that's been a fantastic experiment. And I also worked out about how pigment shifted their wavelengths during the breeding of plants, particularly barley. I then went to Copenhagen, we had a love of Denmark. And then I spent 4 years sequencing 4 kb, 2 years sequencing 4 kb of DNA, about 200 autoradiograms, about 100 base pair per autoradiogram, if you like. This is the computer that we analysed the DNA sequence on. I got a phone call about 2 AM one day. I was to do the assembly at night, because that was when I had access to the computer. I got a phone call, and the guy was from the finance office at Carlsberg. He said, "Do you get paid by Carlsberg?" I said, "Well, I think." He said, "Well, get the fucking off the computer, you've crashed it." So with that knowledge of pigment biosynthesis and sequencing, I was clearly well qualified to become a lecturer in molecular plant pathology. I just had a bit more things to do preparatory before getting going in this subject. And so what I did is I had a conversation with Mike Daniels and John Santos, Richard White, and Tim Hammond, because of some of those names you might remember. So basically, I started out as a lecturer in Norwich, UEA, with the remit of studying plant pathology and using molecular biology. And the question was, what pathogen to work on? And Mike Daniels, who you ought to know, is basically the founder of the Sensory Lab. He was very, very calm, very, very knowledgeable, and he basically advised me. He said, well, biotrophs are much more important to work on. Biotrophs are better than necrotrophs, but you also want a biotroph that you can grow in culture. So we therefore picked up working on tomato leaf mold Transforium fulvum. So I then carried on working in Robert Peter Hunt. This was starting a tradition of mine, which is to get other people to do the important experiments, because frankly it wasn't very good at it. So the interesting thing about Transforium fulvum was that the culture filtrates were not a very useful thing to work on. So Pierre Witz was, of course, the Mr. Man, the king of Transforium fulvum. He had done his PhD, and for those of you who've ever had a bad PhD, you should take solace in thinking that Pierre Witz one was a carp-fish as well. So he ended up concluding incorrectly that the culture filtrates of Transforium fulvum was a good source of important molecules. He then started his lectureship and writing them as well, and developed this technology of taking the intracellular fluid that you got when you grew Transforium tomato, you infiltrated the leaves with water, spun the water out, and that water contained the important molecules. He had by this time a collection of races of Transforium, race 2, race 4, race 5. Race 2 could grow on cultivar with a resistance gene 5, so there was a pathogenic line of tomato as well. So we had all these things, and Pierre gave me these resources to get working on. So there were I had two great ideas. One was to clone the AVR gene, particularly AVR9, and the other one was to clone the CF resistance gene. So this was spoke to grants, got people, and sort of tried to do it. And then this guy cloned the AVR9 gene, this guy cloned the CF9 gene. And I thought, I'll better do something else then. So a third idea, Transforium was a model pathogen. How do pathogens cause disease? Because it seemed to me as a biochemist, we really knew almost nothing about how pathogens actually cause disease. What do they feed on? How do they sporulate? Can we therefore invent novel antifungals and fungicides, antifungal GMs? So for the next 12 years at UEA and then another 3 years back at Copenhagen, we did a variety of things. I just reminded today that Marc-Henri was the first person to discover transposon in the Philippines fungus, and we worked on a bunch of things, including down, which we've been talking about today. Then I got and we continued with a variety of people, including some you might know like Nick Talbot, and people with promising futures called Peter Solomon, who joined me in Copenhagen. So at that point, also in Copenhagen, I was playing around with carbon powder mildew because, as I said, probably the firefighter had more financial clout. So Ken Shirazzo in my lab, there was the first person to actually prove that MLO had been cloned by expressing it in shooting it and finding it. That was work done by Steve Thomas, who was also significant. Now, at that point, I sadly lost my job. I won't go into details of how that happened. I spent a little in what is now Syngenta, what was Dennis Hill, and tried to figure out where my life was going to go with my family and all this thing. So I got the opportunity to go to Australia where there was a farmer-funded project, GIDC money. You've heard about GIDC today. They offered a 5-year contract, and I had 5 people. He was on them to cash out 3 people. And the remit was very clear. Forget about that biotraffic nonsense. We've got it sorted, but network tracks have become really important. So that's what they wanted us to solve. So I had 2 project ideas. One was to create electrotrophic fungal pathogens, but one was to look at electrotrophic fungal pathogens of megacardia. So this was building on a project I worked with John Turner on Arabidopsis pathogen mildew, which should be very successful. So we thought we could try to do the same thing. Arabidopsis really didn't have any electrotrophic pathogens, anyone ever knew about, certainly not the ones that we once was working on. So the idea was to find R genes for astrocytosis disease and rabies. But this is where I started to get worried about this biotrophic, necrotrophic, heavy biotrophic, dogmatic stuff. Everyone said necrotrophic pathogens are not so specific. Well, that turned out to be the case, not to be the case. The pathogens that we recovered from lentils and chickpeas didn't really go very well on megacardia. So eventually we abandoned that project. The project walked into mammary legume crops, Purine Vanderson in the audience was doing that, and effectively mediated the sector of pathogens. So the other project I decided to start then was molecular detection of Staphylococcus aureus, Parastaphylococcus, and the names change faster than the people working on it. And the hypothesis was, if we can understand how these necrotrophs work, we'll be able to design some antifungals, come up with an anti-pathogen GM strategy. Most importantly, I was quite keen to have a job in 2005. So what we actually tried to do, what we wanted to do was take a pathogen to turn it into a model system. I was inspired by the Magna-Porter world and the Saligo world. Our choice of pathogen was really very straightforward. We wanted a pathogen that people knew something about, but which is also important in Australia. So when you work in Australia in the grain industry, you're very excited about how much the disease actually costs. And in the case of West Australia with Phenodora, it was big. But also PTR, Tantalopically. And I'll just put it in here for a sake that it was actually bigger than the rusts. So what did we know about Esnodora? So there's 3 characters here. This is Chris Hayden. He was Father Stagger. He was the person who started doing molecular biology at Stagger Hospital. His student Simon Cutler I was still in contact with, and a student Neil Hall, who was at that time working in Long Ashton in Bristol. So we had these group parties, which Peter will remember, called Brainion, where we discussed things under the influence of alcohol. What we do know about Esnodora, it could be grown in culture easily, it could be transformed. It used to be a major pathogen in the UK and Northwest Europe, but actually it wasn't any longer, but it was in Australia. It was actually the model fungicide species, model species used by fungicide companies. Gene replacement was discovered just before we started in 2000, but there was complex root genetics. So where did Stukenbrock come in? So one of the about 3 genes that we characterised for Esnodora was this gene called Stukenbrock, which was, this is a title of the paper, which this is a typical title that irritates me. The title doesn't include the name of the pathogen or the limitations of the taxonomy of the organisms. So you'll struggle to find this paper. But he discovered there was a gene which he called Stukenbrock. It was secreted in the extracellular fluid, culture filtrate. You remember what Pierre did in his PhD. And it was toxic on all sorts of plant species. So that then we started with the ISCLB. Peter Solomon was very influential. We knocked out genes. We sound a bit like a multi-level, sorry. We won't go any further that way. We knocked out various genes. We were trying to figure out how the pathogen works. We developed an EST library, or whether we gave it to Bruce or Bruce gave it to us. We knew about the Mason type gene, and we've done some work on the cytology. So we knew what was going on at some level, but the time was ticking. 2003, 2 years ago, which brought me to Tunisia and the Tim and Jim moments. So I met Tim at this meeting, and he gave a talk at Staganospiridorum. It was the first time I'd heard someone else talking about Staganospiridorum, in which he talked about culture filtrates that produce proteins that were already purified or semi-purified, induce specific responses in wheat cultivars that could be mackerel. And I said to him, with what I describe as a healthy degree of skepticism, you're talking bollocks, mate. And I thought he was talking about stockbox, so he got very worried, and I'm not sure they were talking about that. So I was basically worried that the PA's culture filtrate error was being repeated here, and where we go. But at that meeting, as you just heard, there was a lot of talk about genome sequencing. When I was at Carlsberg, we just about finished off the Saccharomyces genome, that was 1996. When I got to Syngenta in 1999, I was able to see the Marc-Henri genome that wasn't to be published for over 12 years. So I knew how powerful it was. And of course the Zymer people were talking about it. So I went back to Perth, where it was now, and our midwife in GIDC was a guy called Jim Fortune, and he basically said to me, I said to him, he said to me, why don't you... it just brings tears to my eyes. He said, well, I said to him, we need to sequence this genome. He said, how much would it cost? I said, $500,000. And he said, you can have it as long as you can spend the money by June. This was April or something. April 2004. He didn't work for much longer for GIDC. Didn't go through the process. So by March 2005, we still on meeting, Peter and I went via Boston to look at the genome sequence. And I would point out that genome sequence arrived in my inbox the same day as it arrived in everyone else's inbox. So we had no privileged access to it. It had 109 supercontigs, had 16,000 genes. I then gave a squash it to Peter and then came back to my office. And the email was there saying it's available. I went through the 16,800 genes before going home to see if there was anything interesting. I was getting a bit desperate by, if you remember Megan's talk, 16,751. Because that gene looks very suspicious. It was like toxa transport. So then that led to our meeting where we had found a toxa-like gene in an Odorum genome that previously we found in the transport genome. This wasn't the gene that Tim was expecting to find. We'll talk about that for sure. But by the later that year, we knocked out and expressed that gene. And what we did was also a bit counterintuitive. I infiltrated, or someone in my lab, infiltrated toxa into the current Australian wheat cultivar collection. And so very susceptible, ultra-resistant ones, the red ones are the ones that respond, and the white ones are the ones that don't respond. I asked our statistical guru in the lab, do I need to do statistical analysis to this? And she said, I don't think you do. So we then started sending out toxa to the Australian breeders, and by 2009 we sent out over 100,000 doses of this. And that really saved the day. We got our grant continued, and we've carried on ever since. Now I'd just like to spend a few minutes talking about how to classify fungal pathogens. So the dogma was, as we have been talking about, that necrotrophs were brutish organisms that excreted non-specific toxins and were born close range. And it was all falling apart in my head. Some of you may have seen the paper that James Hayne, again, was also on, called Catastrophe, best named paper I've ever written. But since then, I've been doing some other work where I had to look at the whole fungal taxonomy tree. And I didn't realise that there are 50 classes of fungi, so classes, Diffidioicetes is the class. I've got it written on my wall. But pathogens pathogens are only in 1, 2, 3, 4, 5 of those classes. There's a few in a few others, but basically probably 95% of all fungal pathogens are in 5 of those different classes. There's 129 orders, and they're nearly all in 11. And so if you look at the Gistidiomycetes, the Oligomycetes, Sorgheriomycetes, Bacteria, these two down here, basically they're entirely pathogens. These three, they're pathogenicity within them. And 1/3 of these classes are general pathogens. And these are these 11 orders. And when you started looking at it from a sort of helicopter view, the clear sprays, which we are dealing with, they seem to have a very consistent pathogenic strategy. Cacnodiales, the zymos, they do have one where they're going in through the stomata. And of course the Erysipelas, they have a very clear pathogenic strategy. The Trichostomatina is a very similar thing. So why rusts and Solarigos, as my question to Arnie on Wednesday, apart from Solarigos, why Magus all the Solarigos have a very consistent pathogenic strategy? I'm asking the question, do you actually need to have a heavy heavy biotrope division, classification pathogen? It seems to me, and this is my challenge to you all, is that these 11 orders constitute evolved pathogenic strategies, and we should look at one as a model to the others and fill out the gaps in the system. So, if you don't believe me, then you can read all about it. And so go out and buy this book often and frequently, get it for Christmas presents, and enjoy it. Thank you very much.
Thank you very much. And we will move on to the next speaker, Steve.
I thought Marc-Henri was next.
Sorry, yeah.
Okay, so maybe this one will be more classical, but we'll see. So here my point was trying to do something on genetics, plant pathology, and fungi, kind of take on message from my past 40 years in plant pathology. So one starts that is my point of view important, that you can think of the plant as a nice castle. The plant is there, it has to stay, it's not going to run like an animal. And it has to defend itself against all the microbes around. And you know that for a certain plant there is only a small number of pathogens. Why so? So the fungus, to become pathogenic, that will be able to attack this castle, it has to find a way to sneak in. And I think my interest in plant pathology was, could genetics help us in trying to know how the fungus become pathogenic or is pathogenic? And my inspiration for that was flow work, as you may know. Okay? So it's a guy from USDN, North Dakota Electoral College, and was trying in plant pathology, not in genetics, but he has done a lot of genetic inputs. And that's also an example of being involved in the right project. So he was doing genetics from the flax fungus because he was involved in breeding for resistance. And meanwhile, he decided to do more work, maybe a kind of side project, which became fundamental. So even the right project will become fundamental. So you know, of course, the gene-for-gene relationship. To apply that in real life, not only this diagram that everybody knows. And maybe what is very important is that you have the term avirulent. It became important at that time, so it's a long time ago, and virulent, susceptible, or resistant. And maybe the other thing that was striking me is that, of course, you have, if you do the genetics of the plant, you have a dominant resistant gene, or resistant gene. But if you do the genetics of the pathogen, which is more or less tricky for Melopsora, to realize is more complex than the diagram, you could see that the fact that the fungus was unable to attack the resistant plant was a dominant gene. So that for a long time, being really the drive, okay, here you must have a function behind this word virulence, which was not obvious. I remember when I was a student, when I was trying to talk about the gene-for-gene relationships to other biologists, how to the yeast geneticist even worse, they were always laughing. Why do you call that virulence? Blah, blah, blah. And then you have to spend half an hour, at least 1 hour to explain that diagram. Oh, maybe, okay, come back when you have cloned the gene. Of course, at that time we had no molecular biology for the plant pathogen. If you want to have more interesting things from that, you have a nice paper in 2003 from dogs, who is really deciphering all these studies, and if you're interested, I will invite you to read that. Of course, this is from the 40s. It started in the 30s, but that's really, really a long time ago. And it took more than 50 years to really get the molecular mechanism behind it. And that's been described by Jonathan Jones in that paper in concept. Of course, now the flax gene has been cloned, and the AVR gene has been cloned, and we know how it works. It's a receptor which recognizes the oncoprotein, but you know that. So, if I come back to my own experience, I was a student in the 70s, you know, you need to go and go, and then I became a student in Université d'Orsay, Paris Sud, lecturing genetics, plant biology, plant pathology. It was maybe one of the few places where you could learn plant pathology, so I decided to go there. And we had a lot of nice lab meetings, you know, with drinks, smoking, speaking. At that time, you could do everything you wanted, you know, in the 70s. You can even go through your PhD thesis. And then in the 80s, okay, that's 10 years after, I became a young scientist at CNRS and doing experiments. And I was working on the toxins, so which you may remember that time, and some of you, at that time we had no molecular biology in such fungi. We are just looking at the yeast people, okay, oh, well, how are we doing? But no way for us. And we could isolate enzymes, we could isolate molecules, cellular metabolite, like this one, or phytoalexins. But then how to prove that it has really a role during infection? So you see a small molecule toxin from the right class, and I can say right, that's my background. And there's no molecular genetic. So despite doing some nice chemistry, biochemistry, and things to publish, it was difficult to have genetic approach. I was able to do actually a lot of work to obtain UV mutants which were not producing, but they were still pathogenic. Welcome to the effect of biology. So that was in the 80s. Still, it took 20 years more to find the biosynthetic pathway. So as you can see, there is an amino acid here, so it's a weird PKS and FPS been found by a Japanese group by chance. And they have made the mutant, correct mutant for the Hindi one, and that's understand 20 years later. So at that point, I could not go further. So I said, I will drop that and we'll see later. And at that time, on the right glass field, the idea was how to do genetics with an asexual fungus. That was maybe the main theme, not only in Maniaporte, but also in other species or Zimo. And we spent a lot of time trying to get genetics there in such fungi. So maybe the first one to do some process in Maniaporte was Ajime Kato from Japan, a mycologist. But he was able to cross, in fact, since we know our different species, or different strains from different hosts, and they were mostly, the progeny was non-pathogenic on rice, and the rice isolates were not fertile to that. So he took some chance, so Jean-Claude Nottingham, as you can see here, was able to find fertile isolates 10 years later, one in Guyana and one in Thailand. That's all. Among thousands of strands, you must imagine the Marc-Henri community was big enough, and everybody was testing for fertility. And that was sent to all the labs, so everybody was able to do some process, including your US colleagues that you may know, like Barbara Valent and Forrest Strumland. And then from the 80s to now, people are using these strains and new ones to do process, mostly to clone AR. And you could see here me, Jean-Loup, and Jean-Benoît trying to find a sexual stage in the rice field with no success. So it took some time also to find that all these fertile isolates from the rice plants were coming from southern China. That was published. Here you see the Guitaro, Pierre Gladieux is here. And it took some time to understand that they're coming from southern China, the foothills of Himalaya, which is one center of domestication of rice. And by chance, some of these isolates from southern China came to Guyana, Thailand, and were collected by Jean, just by chance. But now you can get a lot from southern China and China. At the same time, the molecular biology started, so the transformation of the rice blast fungus was obtained in 1987. It was a PNAS paper at that time. Don't expect to have a PNAS paper for transforming your producing neuron. That was Barbara Valent and Forrest Strumland. The first ADR was cloned in 1995, as this book showed. For us, it took us, at that time, 12 years, 3 PhD, clone an ADR gene, starting from the process to really the complementation. So right now you may know that maybe in 1 year or 2 you can clone by map-based mapping. Then of course the genome was obtained in 2005, so in between, during these 10 years, or let's say 15 years, you could do some molecular biology, but you don't have the genome. So you have to do PCR, you have to have libraries, plasmids, BAG, all kind of genomic tools that you will have to sequence, hybridize, just to find a piece of DNA of 1 or 2, 3 kb. So like Richard, I've spent a lot of time sequencing on gems, few hundred base pairs to know where I was in that library and is it a good gene or not. Hopefully, after the genome sequence is published, then the life becomes really about this. So it's evaluated to make oral combination as nice as it is, and just So then for Zymoseptoria, so I moved into Zymoseptoria more recently, but you could take a similar story. So first, Zymoseptoria triticide was thought as an asexual fungus. I'm right?
Into the service and
yes, before. So Sanderson, I think, found some ascospores, and then it took some time also to be able to perform control costs. So that's no work yet, and you can see here the segregation obtained by Gert on Goodwin and Webb with a nice segregation with yes or no for the adherents. And you can see also here they are trying to find the sexual stage in the wheat field with no success, because like rice blasts, I think nobody has been able to find the sexual stage during an epidemic. So maybe Bruce has some ideas on where to find those.
I can find it in any field I look into, and I can teach anyone in this room how to do it if they want to know how to find it. I'll show pictures.
So beside Bruce, it's quite difficult. I think for rice blasts we don't know. Okay. So then, thanks to the genomes being available, that's changed the life of everybody, I guess. So let's go to the start. So Marc-Henri was the first to be sequenced just after Neurospora and Aspergillus at the Broad Institute. So it was an international project with 100 people involved, 500,000. It took years. Then we had Panhalos to Neurospora, the same price. Less people, but better people. So we could see that the Broad Institute was here, it was 13, but treating with the Broad Institute, but And then the other one was sequenced by the Broad Institute. So they're all, botrytis was, and Stereotypia was sequenced, one by the Broad and the other one by the Genoscope. So we had to apply for big grants with National Institute, and it was tough to get that funding. Then, 4 years later, we have much more, 100, including Zimosetoria. So I'm taking the date of publication of not necessarily the availability of the genome. And then you start to have Illumina, which arrived before we had only Sanger sequencing, expensive, big machines. For people who have been visiting the Broad Institute at that time, a few of you, the Broad Institute factory for sequencing was 3 times the size of that room with machine everywhere. And a lot of people. It was like a factory sequencing. So Illumina came in and then it changed also the life of genome sequencing. Then the GTI really came into after the 2002 and was able to sequence, decided we must sequence 1,000 genomes, but almost there. Mostly it's how to get DNA and get people to fill out all the taxonomic groups, because the idea is to sequence all the taxonomic groups, not the fungi which were successful in getting grants to be sequenced, because of the group before. And now we have the PacBio arrive, so the Demosectoria PacBio in 2020, few thousand euros. And now we have the new PacBio, which is only 300 euros. So now you don't need a big consortium. You can sequence your genome perfect, fully assembled for a low cost. So of course, I don't count anymore the number of genome available. It's over the roof. Okay? But that's now feasible for everybody to use genomics. You don't need these big consortium. I think that's changing the line. So what next? To me, it's okay, we now have much more data. And the question is how to analyze them. So we have seen software, how to, or let's say improve the annotation. Everybody's been involved in that, so it's going to be improved again. So that's a question mark. That's still because of all this data we can, and all the evolution of the techniques, we are able to study more easily the complex systems. Genes and interactions, still we are limited by the phenotyping. So let's say STD16 cloning, most of the time spent is phenotyping. And we need to model all this biological process, so that's still not very well developed. And hope maybe artificial intelligence will help, but we need to model now because you cannot rely just on the gene-for-gene relationships. You have to think of all the interaction and make models. And then with these models, maybe we become and enter the era of predictive biology. That's my goal. Thank you.
Thank you very much, Marc-Henri. And then now we'll move on to Steve Goodwin.
All right, great. Yeah, thanks for inviting us to speak here at the what's been very referred to as the retirement session or the 501 guys session or the dinosaur session, as someone said it was. So I'm happy to be one of the dinosaurs up here speaking to you today. But first of all, Gert mentioned the green flash. How many people have seen the green flash? There's one. Good. Hey, you guys can get out more. I tell you, it's real. It does exist. We did not see it that day. All right. What I want to do is tell you my personal journey on how I got where I am now. Some of these fields and what we did in the old days, what it was like, how hard it was before we had all the resources we have now, and how we got to where we are now. So let's see. So I'm going to start with when I started grad school. 1982, I go to California, I grew up on the East Coast, never been to the West before. I go to California, the first person I met there pretty much was Bruce. We started in the same lab, the Bob Allen's lab at the University of California campus in September 1982. And I don't remember who got there first, but we started within about a week of each other, right? And I remember getting very little guidance. Bob Allen, he retired a couple of years after that. He was a famous plant breeder, very enthusiastic. He had these weekly meetings with us. He discussed all this stuff, but not much about our research projects. At least that was my impression. And so he gave us his grant proposal. He had a grant proposal on multi-lines, which of course now it sounds like Lisa's got actually working in Denmark, but in that case it was all the rage. And said, here's this grant proposal, see if we can get a couple PhD theses out of that, right? So part of it was to look at these multi-lines made from barley cross populations and inoculating the races of pathogen, and we divided it up. Somehow Bruce took the host side and I took the pathogen side. I don't remember how we decided that, but somehow it came to that. I started on this. We had Bob Webster was a co-advisor on this, a plant pathologist, and he was always a little skeptical. I don't know about this pathogen stuff, and I thought, well, you just haven't seen me, There were other grad students who probably wouldn't work, but I'm going to work so hard. I'm going to get all this data. The arrogance and ignorance is something else. But and I worked on this for like a year and a half or 2 years, and I realized it's not going to work. I was supposed to inoculate these field populations. Bruce looked at the yield and all stuff on the host compositions, and I was supposed to take these. Inoculate these 4 pathogenic races. I was supposed to phenotype them on 21 differential cultivars and look at the changes in the frequencies of the races over time. The first 100 isolates I analyzed, I got one that was like one of the original 4, and the others were all different. I said, oh man! And so I took them all, stuck it in the folder, forgot about it for 5 or 10 minutes. Eventually I realized, I looked at it a different way. I finally did publish it, but it took a while. Very frustrating. And I said, maybe do something else. I need to develop some markers for doing something. The lab was big population genetics. They needed isocymes in barley and maybe oats. And I said, I should develop some kind of a marker system for this pathogen. Back then, as far as I know, this is the first paper developed by Philip Steve at UC Berkeley. Was the first paper that I know that mentioned fungal population genetics, 1975. Okay, and so in those days, if you wanted to keep up with the literature, read one paper and you're there. Pretty easy. It's like, oh man, so I kind of keep trying. And these are some of the jealousy we had. The data sucks, right? But he still did it on paper, and I figured I need to do something. Much better things in bond, and I figured I'm going to see what I can do. So I went and started screening things. I mentioned one part about how tough it was being a grad student. My major professor didn't pay a penny for me to do anything. I don't know if there was a fellowship involved. I remember Bruce was on a fellowship, but I wasn't. I was a reader or a TA for 11 quarters. And then I had a fellowship from the NIH, National Institute of Health, on a training grant. I had to pay back afterwards if I didn't do it. It was paid $450 a month. It sucked even then. Because I worked in the Department of Physical Medicine and Rehabilitation as a computer program. And I did a lot of computer stuff when I was undergraduate too, that I'm going to talk about. And when I went to meetings, I paid my own abstract publication, I paid the registration, I paid the travel, all myself, not a penny. I don't think that's right. That's why my lab, if you're grad student, you get paid to go to meetings, because I don't think that's right. My major professor didn't get it's bad. I thought that was the normal way, right? But we were doing this stuff. Anyway, the other thing was there were no programs for data analysis. If you wanted to do it, then you had to do it by hand at a calculator and write your own programs. So I used to work as a computer programmer. I tried to talk myself, Steve, wrote a lot of programs for doing data analysis. It was a very different way of doing things. I also had to go, I screened a bunch of isozyme systems to develop them. And back then we had to go to libraries. You guys probably don't even know what a library is anymore. Right? A library was like a casino for academics. Because you go in there and there's no clocks, there's no windows, and you read stats for hours and hours, and you find a reference to something, and you go look it up in some other thing, and find that reference, and go look that up, and you have these bound volumes of journals, and then you take the bound volumes, pocket pull up quarters, go up to the photocopy machine, and type all your stuff, or you can check them out and run them all the way across campus to photocopy. You're in a different world. So I did that, I had filing cards full of all these recipes that I tried out. none of these nice designs. It worked out very nicely. Then she got a PhD thesis out of it, but it wasn't the one I thought I was going to do when I started. It all worked out. I want to talk about IT stuff, you know? All this computer stuff we're doing now. And Alice gave a great talk this morning about machine learning and all these sort of things. Back in those days, I took a computer course when I was an undergrad at Duke in 1979. These are my programs from 1979, okay? These are the cards. Each one of these cards, you can see here, there's a little line telling you what the code is, and then you would punch it. You had these punch card machines, right? You punch out your cards, and then you have this big stack of cards that, like this. You take your cards, and you go way down the hall to a card reader machine. It was this huge thing. You put the thing in, go, read all the cards, and then the computer was in this whole big room full of the computer, right? And probably those computers, the supercomputers in those days, were probably far less powerful than what a cell phone is now, right? But you go there, wait for the thing, and sometimes if you're working there late at night, all of a sudden you hear, no, I can't believe it, music full of my side. And you realize, oh, somebody dropped my cards. They were numbered, so there's no way to put them back in the order. So you do that one time, and then after that, you see people carrying these cards down to the meter, like more like a newborn baby, or they're not dropping it again. It was a different world. And this is my first portable computer. This is an Apple something, I don't know what they called it in those days. This is a MacBook Air from 10 years ago. So it had about the same footprint, but look at the size of this sucker. This thing, it weighs a ton. It's sitting in my office right now. I've never seen it. So I took that on a trip one time and said, forget this, I'm not lugging this thing around. The only good thing about it was you didn't have to go to the gym. After all you got your workout for the day. This is when I was a grad student, this is part of the Allen Lab, this is all the gel writing rigs and that sort of thing for those times. We worked all the time, that's what I remember, but sometimes we've been out of the lab. So here we are, here's our field project. I think that's Bruce on the back of the harvester with a big barley field there. Oh, this by the way is Brian Stephenson, I don't know if anybody knows him. And here we are, we climbed to the top of Lassen Peak. This is 1986, right? And I grew up in the East Coast, we didn't have tall mountains, so I didn't know this the whole concept of it. It's really cold at the top, right? Bruce obviously knew this, he came prepared. He was seeing then he did that, right? This is John McDermott, he was prepared. And what the heck am I wearing? I never saw that. Oops. I never saw that. Yeah, it's so bad it really screamed, I guess. But I never saw that sweatshirt before. I must have borrowed it. Check out these shorts. I mean, that was in 1982, right? They were these short shorts. Real men wore shorts that were short. Nowadays, we have these long, girly shorts. You know, I don't like that. Shorts, they're long, right? So, I told my girlfriend, I had to go out and get some long, shorts like that. She said, you go right ahead, but I'm not going to be seen in public. I'm not very supportive. Anyway, and I was going to tell you some of the story conferences. The first one was in 1976, when I was still in high school. The second one was in Montana in 1983, when Bruce and I were in our second year of grad school. So, one of the other things we did, we went to Death Valley. I grew up on the East Coast. And the West Coast was the desert. So I've never seen a desert before. Let's go up there. Every year for spring break, we go to Death Valley. We have quarters, we have March, and a week or two we go out there. And Death Valley, we take pictures. And I was going to do photography. I got a camera. Bruce taught me how to use it. And everybody was doing photography. Joe McDermott was doing photography. We go to Death Valley, and there's this big sand dune park, this steep flat sand dunes in the middle of Death Valley. And we go there at sunrise, and then we all split up and say, okay, we'll be back here in an hour and a half, right? And they're going to go off, shoot their pictures, and this is one of mine from 1986, and I shoot this kind of thing. Broad, big vistas, taking in the sand dunes, the big sweep of the whole thing. In those days, we all shot film, and they were slide film. And so you'd have these Kodak mailers, and you'd shoot your film, you drop it in the mailer, to the mail, and 2 or 3 weeks later, you get slides back, and then you meet somebody's house. Not like Instagram nowadays, you post them all. It was very social nowadays. You'd actually meet in people's houses, not through social media, to talk to each other. It was totally crazy, right? You gather at somebody's house with a projector, and then you watch the slides. And it was very interesting to do this, and so the pictures were completely different. I had these sweeping vistas and all this sort of stuff. Bruce had focused in on like almost macro photography, the little tracks of insects through the sand, and the ripples of it, how the light played on the ripples of the thing, and then there's a leaf that had blown something, had some other interesting kind of, I didn't even notice any of that. All this stuff. And Joe did completely different pictures. Bruce is going to talk about a little bit more. And here's another thing. I was over back in April. My girlfriend's in Goodwin and wanted to go to Death Valley and see the flowers, so we went in April. And I still take the same kind of pictures as I did. That big, strong business. And then here's another one from Death Valley in April. The thing I want to point out that I learned from that, from seeing all these different perspectives, same place, same time, same type of thing doing photography, pictures were completely different. They all had sand in them, but other than that, completely different pictures. And what that taught me is, when you have a team to work on something, the more diverse viewpoints you get, the more people look at it, everybody's going to see something different, everybody's going to know something, so when you have all that together, you get a much more clear picture and a much better product than if you try to do something by yourself. All right, that's that. For postdoc, I want to go do work on potato leaf blight disease, right? And I wanted to go there and work with Bill Fry, because it was the other side of the country. Cornell University. And he could do population genetics with molecular markers, and he worked in Mexico where they had a lot of diversity, right? And so the third Sartori workshop was right after I became a postdoc. And that was here in Zurich. So I went to Cornell, and the first thing Bill Fry says, glad you're here, want to do population genetics. He was an epidemiologist, didn't really know anything about population genetics. He said, but you have to set the lab up from scratch. There was nothing there. I had visited it. I had no idea. It's like, oh my. And he said, but I got a lot of money to do it, like $5,000 or $6,000. I'm thinking, oh no! If you had $150,000 maybe, but $5,000, you couldn't get scratch here. And if I had another offer on the table, I would have said, I'm out of here. This isn't going to work for me, you know? But I didn't, so I stuck it around. I developed this thing. The other thing was, a grad student said, I already claimed all this stuff from Mexico. It's like, yeah, we're kidding, right? And so I had to work on populations there. Well, leaf blight was controlled by metallitis. There weren't any epidemics to sample because it was wiped out with the fungicide, right? But there was one epidemic, I collected it, and 30 or so isolates. They were all identical. It's like, oh man, genetic differentiation. It's like, there isn't any. What am I going to do? Something else. And I was talking to Michael Woodrum at Cornell, and he said, you should do, what about DNA fingerprint? And this guy, Alec Jeffries, had served Alec Jeffries to newcomers. He was 90th for this, actually. And he had discovered a DNA fingerprint in humans in 1984, published in 85, and this was 1987. So it was pretty early days, still a DNA fingerprint. He said, okay, I should do that. So I developed a DNA fingerprint probe. Okay, 5 words, took me like a year and a half or 2 years to do it. And there was a lot of screening. Back in those days, there was these films of radioactivity, P32, and all that, southern blocks, all this kind of junk. Took forever, major pain. Anyway, long story short, finally did it. It turned out there was a single clone spread out over the whole world. So, kind of interesting, but really boring if you wanted to look at genetic differentiation. I still got a PNAS paper and several other papers. And at this point, I had quite a few papers. I got 4 or 5 papers out of my thesis ultimately. Some of them published in Manual actually, but I don't know, 6, 8, 10 papers from my postdoc at this point. So I'm applying for jobs, and the job market was very challenging. It's not exactly the words we used at the time, but it was very tough, you know. There was tons of people looking, and I started getting rejection. Sometimes you get 275 or 300 people applying for one job. And I'm saying this just to give a perspective of what it is. And in case you did, this is what I had when I started applying for jobs, right? I literally applied, I counted once, there was 100 jobs. Look at odds of that. Exactly 100 positions I applied for. I got 99 rejections. I got 1 interview, and then I heard nothing. and I still got that file in my office. If anyone wants to see it, I'll show you my 100 rejections. I'm 99 rejection rate. Then about a year and a half after that, I heard that, the person's position was frozen by the USDA, then they re-unfroze it, and they offered it to another guy. And then he turned him down, so I got the consolation prize. So, out of 100 jobs I applied for, completely rejected by 99. 1, yes, but I was the second choice. So what the take home message from here is never give up. Don't worry about it. If you're not getting rejected, you're not trying hard enough, keep trying. Okay. So then I went to the USDA to work at wherever, University in Lafayette, right? And so I always had to be a septoria pathologist, or a cereal pathologist again, on septoria. And Bruce, I called up Bruce, and he said, if you want to get a fast start in septoria, come on down to my lab. I think those are exactly the words he used, something to that effect. I said, sounds good. So I started, I think, June 12th, and I think it was 4th of July weekend. I went down to Texas A&M to visit Bruce's lab, and one of the first people I met there was, let's see, one of the first people I met there was, I think his name's Gus. And he comes up and said, I'm Gus. I smell so pot every day. I thought, congratulations. After that, anyway, kind of lab situation with Bruce. I don't know, Gus wasn't on a good trajectory in my opinion at that point, but he probably ended up being a CEO of Fortune 500. Bruce should have said that. Anyway, but he did introduce Joyce Lee Sharma, who was decided to help me out. And Josh Lee was not a pothead and was very dedicated and very helpful, and he spent 2 or 3 days working with me, and I was really delighted to see Josh Lee here today. I'd seen him about 20 years ago. Thank you, Josh Lee, for helping me get going almost 30 years ago on Septoria. The other thing that happened is that USDA would give us 5-year plans, and I had to write mine up. And so I did, and what I was going to do, we talked about earlier, Sanderson had discovered the sexual stage for, sexual stage for Marc-Henri Lebrun in 1972. No one figured out how to do it or cross it. And so I had a 5-year plan, I had written it up on how to make crosses, all this stuff. I ordered these special integrators to alternate the light and the humidity and the temperature and all this junk. I was going to do that, and I hadn't started yet though. And I went down to this meeting at the International Potato Center. There was a guy at Purdue, Tom Hodges, and he didn't work on potatoes. He said, hey, I got this all-expense paid trip to go to the International Potato Center for this potato thing. He said, I don't work on potatoes. He said, last time I went to Peru, I got food poisoning and almost died. So you want to go and get me? I said, well, the food poisoning part I can kind of do without, but that sounds good. So anyway, back in those days with USDA, now it would be almost impossible to do it, but I was able to get permission to go. I go to Peru on this all-expense paid trip, and I'm talking to Bill Fryer about what to do. I'm telling him, I got this project, I'm going to start making a process, blah, blah, blah. He said, you've done a sabbatical at Goodwin. And he said, I'm just talking to Corvette stuff up there, and he's got a student who just nailed that. It's like, really? I guess I better talk to him. So we talked about this, and then by the time I got back from Peru, I had an email from Gert Kema saying, oh, by the way, I heard you want to do this, we should try to collaborate. And as Gert said, then I went to visit his lab right away. And then here's the picture of here, and this technician Elsa. I wouldn't recognize that picture of her up there, but and Elsa spent 2 or 3 days with me there, and this is June of 1996. And like I said, it's easy in those days, no paperwork, all the stuff we do then. I did get an import permit, and so I got going through U.S. Customs, and I had this permit. I said, I got a permit to import these plant pathogen isolates. And the guy said, what? It's a permit. He said, oh, wow, a permit, I can't believe it. And it's like, hey, Bob, check this out. I actually got a permit. I said, nobody ever does it believe it. Okay, so they didn't even look at the box, right? Just like, I need to go. Okay, I did a little bunch of vascular resistance machines and that sort of thing, published a few papers, and I don't know much about that. What I did want to say, I did that in part because going to meetings, as Tim has said directly, is that you go to meetings, you meet people, you build collaborative projects. I was able to map 5 resistance genes, I think, without making a single cross myself, by getting things, we're doing collaborations with other people. Collaborations are good. Don't try to do it alone. They're much farther, much faster, work with others. Now, the Septoria meetings coming here. I'm trying to explain how we do stuff. So the 5th one was in Mexico, and I think Gert, me, and Marc-Henri were all there. The speaker on Wednesday talked about weekly coaching, whether that the guy who works on the corn smut. I had never had it before, but you go to Mexico, they had a big vat of this at the reception, right? A big bowl, and it's this kind of black, gelatinous goo. But it tastes really good. It's nice, mushroomy flavor. So we were coaching Good, very good. You should try it. I'm sure you've had it, right? Yeah, very, very nice stuff. Okay. Now, after that, we had to decide the meetings. And in those days, not just this meeting, they had meetings were decided democratically, not like a elite committee that does it nowadays in back rooms, right? In those days, you have 3 or 4 people come up with proposals for the next meeting, and they give their thing, well, go to this and blah, 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 and then everyone vote on it, right? And so we had, I think the choices were Czech Republic and somewhere else, and I don't remember. And one was Tunisia, right? Moncef Arabi gets up there and he says, okay, show pictures of Jebel Island, women in bikinis, and he said, belly dancers. Where he said, beaches, belly dancers, and bikinis were there. In those days, the meetings were predominantly male, as you can imagine. I think there were probably, it was Christina Keller, right? And Lisa Gildas and Suman, and probably Lisa Jorgensen. That was it, you know. Oh no, a bunch of guys. So get a bunch of guys together. We're not proud of that now. I apologize for that. It really was a bad thing, but it was good to get a bunch of guys together on one chromosome we can't help ourselves, right? So anyway, things are much better now. Like I said, from Death Valley, diversity is good. All you really get involved. Now we get much more progress, much faster because of all the diversity. And that was last millennium. Now that was a whole world ago. It's a completely different world. So we're going to do genome sequencing. We've heard part of the story already. To do the sequencing, Eric flew to my office. He spent a week in my office we were writing this proposal. And it was right before the Tunisian meetings. I had a guy at NIH lined up to do the sequencing for the National Institute to help with the sequencing. And about a week before that, he calls me and says, oh, I'm doing the budget, and USDA only pays 11% overhead. Yeah, that's right. He said, we need more than that. We need like 50%. I said, they're going to do it. He said, do they negotiate? No, they don't negotiate. It's 11%. So I can't do it. So what do you mean you can't do it? Oh, I'm backing out. You can't back out. He said, oh, sequencing is no big deal nowadays. Just submit it without the sequencing center. I said, you can't submit a grant proposal to sequence the genome and say, oh, give us the money and we'll figure out the sequencing part later. That's not going to fly, right? So I went to Tunisia and Gert found out, and I said, I guess you're going to do it. And Gert said, no, we're not giving up. We got to keep doing this. I said, what do you mean? He said, well, we have to call people from here. I said, how do they call from Tunisia? And he said, I got a cell phone. And I said, what's a cell phone? It was cell phones in like the 80s and 90s, like these big nuclear powered things. People talking to people about brain cancer. I think that's true, actually. You need to look it up. But don't have a cell phone. And so he called me. Can you help us now? Try to get this guy. You have his phone number? Oh yeah, okay. I called another one. Oh, get his phone number? So I don't know how many people we called. Gert, like, I was going to give up, but Gert would not let me give up. Now he kept saying, trying to call this guy. Finally, after all this time, let's see. We got a hold of Dan Robson at the JCL. And he said, oh yeah, I can do it. He said, kind of a short timeline. I think we can make this work. Send me the proposal. I'll work on it. And all you have to do is go back, finish off the budget part, and then put it together, get that sucker submitted. I think we were 2 or 3 days after I got back from the Tunisian meetings. And I get, pardon?
Just one.
Yeah, well, yeah. Anyway, so I get back, and what I hear is nothing. And I call him, nothing from Dan, and then the deadline passed, we missed it. And I had a talk at Tunisia talking about plan A, plan B, plan C, and we did it with plan F, right? And Dan called me and said, sorry, I had to attack of kidney stones. I never had those. It's the most excruciating pain you could ever have. So he was going to do it. He said, but have you ever heard of the community sequencing program? I said, no, what's that? So he said, oh, this is a new program. JGI sequences some human chromosomes. And they were done, and they had the sequencing facility, but nothing to do. I said, let's open up the scientific community. I said, okay, good. So we submitted, and we already had the proposal written by the community sequencing. Now it's a community scientific program. It's really an excellent program, by the way. It's still going. And you just submit all the scientific reasons. You don't have to bother with the budget. We have that done, so we submitted that in the first program through that. Yeah, and so then we got that done. We got the sequence. Then we got the sequence, and then it's like, oh no, what do we do? We got to annotate it. Because we don't know what we're doing, right? We're always like, wow, the next level of ignorance up. And so when JGI said, oh, we have to have a meditation jamboree, it was excellent. What's meditation jamboree? And so we said, oh, we can do that. So we did. We divided up the whole genome, and everyone who was interested in certain genes took or wanted to learn about them took, I'll take these ones, and you were supposed to work on them at home for 2 or 3 or 4 months. I don't know the time frame. We get together for 2 or 3 days and make reports on it. And all the other thing is we worked with the program people. Computer programmer can think around all kinds of programs to do really good stuff, but it's not useful at all to you unless you get some guidance. It's really frustrating for you and them, right? But this time we had the programmers there, and here's the education jamborees. And they're telling everyone, if you have a tool you want to do, the programmers are here. Let them know you do this. And several times people say, it'd be really nice to have a tool to do this. And the next day we'd say, oh, by the way, we took that, here's this, so very, very nice communication. Anyway, that's the story of how we got from there to here. Here's my personal story, much more complicated than that, but I didn't have time to talk about a lot of this stuff, but I put marks of where the tutorial meetings so I can see they get more frequent. I forgot the 2022 one. And for me, beyond is going to be, I'm doing a lot of work on tar spot and corn now, which is an omega pathogen and bit of pain, but a very serious problem in the U.S. And I'm not ready to join the dinosaurs just yet. I'll 3 to 5 years, and then I'll head out to the beach. That's all I have. So.
Steve, you can have another career as a stand-up. You showed one of the many skill sets there. A great future for you beyond the entire stand-up column scientist. Certainly, dream out.
Good to know I have something to do in retirement.
Yeah. Okay, so I was trying to bring in a bit of humor, but I won't be able to match some of the expectations at the right level in the beginning here. But what I want to do is just give a quick 15-minute, 20-minute summary of my personal journey, which has gone on for about 42 years now, going out as with the other speakers, I reached back to the PhD days, and let's start with that. So that first stage of the training was at UC Davis with Steve. I think we started about 1 week apart. Yeah. We had 2 supervisors. One of them, Bob Bludster, was a plant pathologist. The other one, Bob Allard, was a population geneticist. Here's Steve, by the way, looking shorts off for this picture. Usually walk around with shorts in the lab too, but they make that looking more respectable. And Joe McDermott sitting there looking very serious because he's not that serious. But anyway, here they are doing work. So sitting on the harvester, catching data from our field experiments. And as Steve already indicated, our package of experiments at that time was mostly in the field with this barley scald pathogen called Rhizospermum cicalis, not called Rhizospermum comuni. I think Pedro Krause gave another name recently, but anyway, it's just called Rhizospermum corn. Yep. So, as Steve already said, we were doing field experiments, but it was Steve who invented the technology which we eventually were using for the population genetics, which was to get these isozymes figured out. So Steve invested many years of effort into giving us a genetic tool that we could then apply. And we had, as a result of work in Bob's lab on barley, we had a postdoc there named Richard Jorgensen. And he was actually into developing molecular genetic markers in barley. And he actually had RFLPs going in barley. And that reminds me, I distributed around the room some of these autoradiograms, because for you younger folks, I want you to know what data used to look like. This was the advanced technology in the 1980s, RFLPs. I have thousands of these things in my office, by the way, in case you are looking for them. And with Rich Jorgen's assistance, we were able to get the first DNA-based genetic marker system working in a Ferroventus fungus, and this is RLPs, the ribosomal gene complex in the Rufus calis. And taking together these different genetic markers, the isozymes from Steve, those who work in the RLPs, we were able then to assemble vast quantities of strains of this Rufus fungus from a single field site and two different composite crosses. I want to tell the composite cross story, but there were two of them. And we did everything the wrong way. First of all, did the experiment, collected the giant data sets, and then went to the literature in order to compare our findings with other people's findings, and discovered there were no other findings except for field C, for and for me, that was like opportunity knocking. I was like, fungi, that's a pretty big group of organisms. Population genetics, I learned how to do that. I think I should start going pop gen and fungi. It just seems to make sense. So essentially, this actually was that first insight that there was a huge group of creatures that hadn't been studied in this way before, and that served to find this reproduction right there. So opportunity knocks. I want to show you the first figure in one of my first scientific papers that I created. I can't remember what the software was that I used to make that figure. But this shows the distribution of the 5 different genetic markers we had in these 1,300 strains. And this is their physical positions in these different plots, practically those composite crosses. And I was so excited to see gradients. This is like this black multi-locus genotype was, really a high frequency here, low frequency there. We didn't really know what it meant because in those days, Rufus Boy was a purely asexual organism. We were taught that it never had a sexual date in its life, and now we know that that was all wrong. So this is actually a combined fungus that has plenty of sex energy. But that was the sort of figure I was proud of back in those days, and was allowed. Now, we did have some fun working there. Some of you might recognize some of these people. That's really easier to see. Steve talked about going to Death Valley, and theoretically we were searching for wildflowers. The truth was we were just out there to have a fun camping trip and do the things that young men tend to do. In this case, we're outside of a mine shaft at the Death Valley, and we decided we all needed to have sunglasses and a hat. And we all brought our girlfriends along to work the cameras. So we had 4 guys, 4 girls, and 4 cameras out there that we work. And we called these pictures the desert classics, we were about to see, because at that time when we started sharing each other's photographs, as Steve said, we all had different perspectives on the different pictures of them. We decided at that time when we were like young 20s, this would be the sort of thing you bring in at a retirement party. Not as I get retired, I'm damn close, but it's time to show the real desert classics. Four young scientists walking off the wasteland. I don't know if you can get it, but that's Steve right there. John's fine, he was working in a different lab, but it was part of a social circle at that time. And as we were taking these pictures, remember our ladies are actually working the cameras. We set it all up and they push the shutter. But one of the ladies got very nervous and scared. This is a national park, by the way. There's a pretty busy highway. Not so far away from where we're taking these pictures. And one of the girls, I think it was John Smichael's girlfriend, this is John. Ran away screaming, "We're going to get arrested! Sexual deviancy! Oh my God, we're going to put permanent criminal records!" And we didn't see her for 2 or 3 hours. She really didn't want to have anything to do with it, so we wanted to keep going. So that was an interesting contrast in fighting. Let's get a side angle. And there's also black and white. So there's many different positions we were trying out with our cameras. We were exploring that photography as it should be explored. And that was what we did back in the PhD expertise. So that was the first stage of my career. The second stage was when I then moved over to Texas A&M University, where I was an assistant and then an associate professor, so half-squared. And that's when I started getting into Sartoria triticite and Sartoria indorum. And that's where we started out with this basic approach of doing what we call hierarchical sampling, the same basic sampling strategy, applying it to naturally infected farmers' fields of these pathogens around the world, and developing these RFLP markers. I got thousands of these things, by the way, so if you want to take some home, just let me know. And as Steve already said, we developed DNA fingerprints based upon hybridization to what we know are rejected elements or transposable elements. And we also did individual loci. We did these DNA fingerprints used for Sartoria triticide, used for what we call Sartoria indorum back in those days. We looked at nuclear genomes, and here's the mitochondrial genomes of the same isolate that you see up here, which is so diverse for the nucleus, for the mitochondria. It's very boring. Where for Sartoria indorum, at least there was some mitochondrial diversity for that organism. And I was so glad to hear this talk today from Daniel's group that there's, yesterday, looking at mitochondrial diversity, because I think there's a sort of, just looking at why is there so little diversity in the mitochondrial genome of Sartoria triticide. And the other thing we started working on was this pulse field gel electrophoresis technology to separate the entire chromosomes of Sartoria triticide in this case. In this case, it's a bunch of isolates from the same field. So from one field, we have this incredible karyotype diversity, including a bunch of these small chromosomes. We might call these accessory chromosomes. These are the ones that go or disappear according to the strain, and the large chromosomes up here, we might call them core chromosomes. But this was the first indication to me that in this fungus, there was enormous diversity, not only for the standard sort of genetic markers, but even for the cytogenetics markers. So this was the first insight into the population genetic structure and phylogeography of the pathogen that came during that time. And I want to say a few words about one guy who all of us are talking about sort of history here. You know, a big debt to this guy, Zahir Iyal, who sadly died of cancer in 1999 at the age of 63. I'm 63 right now, and I feel a certain resonance thinking about dying at this age. I hope I don't. But anyway, he gave us this great book on how to do work with Sartorius, triticide and indorum. And though he did invite me to his house to have lunch when I visited him in 1988, soon after I became a professor in Texas, he would never send me infected leads from the wheat fields of Israel. And it goes on for 3 years, and I finally gave up and found a young PhD postdoc who had just finished his postdoc at UC Berkeley. He was going back to Israel to become an assistant professor. His name was Oded Yarden, and Oded did exactly what I asked him to do. He came to that collection, several collections of infected leaves, sent them to Texas, so I can isolate the pathogen and do our RFLP thing. And as I was hoping, that became the key to unlocking the evolutionary history of Sartoria triticide, because that's how we got the first evidence that this pathogen came out of the fertile crest. And so we found huge diversity from mitochondrial genes, everything was super diverse there. And so that was a very important question. Now, at this British Society of Plant Ecology meeting in 1994, the whole meeting topic was population genetics and necrotrophs, and Zahir gave a lead-off talk for that meeting, talking about his work with Sartoria triticide, and he was Mr. Sartoria to me, and to you as well. He was the guy. He knew everything about it after 25 years of research on it. And in his talk, he gave it the sort of paradigms that were in the discipline at that time. Sartoria is mostly floral. The population is separated by different 10 or 20 kilometers, are different from each other. There's very little gene flow, and it's all stubble. So everything's coming out of the soil and out of the infected leaves. And then I stood up and gave my talk, and based upon large data sets, essentially tore down all of these little paradigms. So I said, no, this system is highly recombined pathogen. As a matter of fact, it has huge gene flow, the populations on different continents are really similar to each other, and it's all ASCIL score. These various scores are super important. And then in my talk, I showed the first data from Israeli populations. So he was sitting in the front row, and I saw his eyes get really big. After I finished my talk, he put up his hand for the first question. And he said, "Bruce, are you trying to tell me that this fungus, which I have worked with in Israel for 25 years, are you trying to tell me that it has a sexual cycle here?" I said, "Yeah, it's been under your nose the whole time." And the audience was, half of them were chuckling and half of them were shocked. But for me, this was the paradigm shift. This is when a lot of people began to get into the idea that, oh yeah, it's probably a pretty sexual organism, and maybe, by the here's what they look like. As I shouted from that room, I can teach anyone in this room how to find it. You can find it in any leaf field where the disease occurs. Just go down to the leaves that are on the ground, they're all dead, hungry looking, find the old lesions, and you'll find a bunch of pseudomonas. It's pretty straightforward. Hundreds or thousands of different experiments, and that's what it's for. Okay, so also we had some fun, and here is a super technician, Rodney Petway, dressing his finest. He looked pretty good in ladies' clothing for a Halloween party at that time. I did have a fairly socially active lab, let's say, quite a bit of merriment going on. As a matter of fact, as the chief marinator, I decided it was time to add quality beer into the college stage environment. So I opened Brazos Brewing Company, and basically made some fine beers, including my favorite, the Texas Sunset Amber Ale. But that foray of mine into business only lasted 2.5 years, which is when I ran out of money. The Brazos Brewing Company had to close. And that's when I decided it was time to leave Texas. They don't like my beer. If they don't like my beer, then it's time to get out. So that is essentially what brought me over to Switzerland. And when I came to Switzerland, I had to teach plant pathology. I'm a professor of plant pathology in Switzerland. And to teach, you have to learn. Well, I had plant pathology classes, and never had the need to actually teach a whole package of plant pathology courses. So I read a lot and came up with ideas of what to teach, and this little gem from the Fungicide Resistance Action Committee, published in 1998, was a very interesting booklet in which they laid out sort of themes for how to determine if fungicide resistance would emerge and how to stop it from emerging. And one figure caught my attention, and still does catch my attention, because they tried to quantify the risk with a numerical scale. But it took 3 different categories, low, medium, and high, gave them numerical values, 1, 2, and 3. And then on one axis, looking at chemical risks, mainly active ingredients and targets. On the other axis, looking at biological factors of the pathogens, and sort of try to delve into the probability that fungicide resistance can emerge. And I read that and thought, wow, what a great idea. And as with all scientists, if you hear a great idea, the first thing you do is imitate, copy, duplicate. And so I took that idea and expanded it out a bit, looking into these different evolutionary forces, the reproductive system, the amount of gene flow going on, the effective population size, and kind of mapped in to it these different pathogens to see if we could look at evolutionary potential of pathogens in that way. And what I want you to take away from this is this great feedback loop between teaching and research. Your teaching can give you the ideas that will then feed into your research, and through your research you can pick up new stuff that will inform your teaching. So if you're an academic scientist like me, then this is the positive feedback you really want to get going. and I took many ideas out of my teaching, which became research projects as well as sometimes publications. So during the time then at ETH, where I'm still at, here as well. So that's where we sort of finished the phylogeography of those pathogens. We worked, of course, with Cereal and Borum, and finished off working with Borum, also with Paloceracemes as systems to be respiratory slide, the right sheep-like pathogen, and of course, the circulatory granulus triticide, this weak blast pathogen. Figured out where they came from, their origins, how they moved around the world. We have done all this evolutionary ecology stuff, QTL mapping, after Gert taught Marcello Zollner in my lab how to do megacross, process good strings. Thank you, Gert, for that. And nowadays we do a lot with GWAS, of course. And as someone said at some point, phenotyping is hard. Phenotyping has become really easy. Phenotyping is still hard. And that's why in these last couple of years I've been investing a lot in the groups that are working on phenotyping. So we're trying to get the phenotype being developed, and there's some nice talks on that earlier already, just to see what we're happening in class. So, and then Daniel Cole really cracked open the population genomics. The others had already made a start there between Eva, so we were already doing that path. But now, I would say we're doing much more population genomics instead of population genetics. And of course, while in Switzerland, we did all kinds of nice social engagements leading up in China with Richard Oliver and Eva McDonald, over in Mexico with Tim Friesen and Megan and Patrick Bruner. You already saw this picture. I guess several people have it, so a nice trip in San Diego. You didn't see the green flash that time. By the way, I grew up in California, I've never seen it either. So, there are by all kinds of nice Google pictures. You Google it about the picture, people did see it. It turns out in special atmospheric conditions to make it happen. But it's been a great trip. Great journey, and I just want to finish with this one lesser-known story of toxin. Everyone knows that toxins are a very favorite molecule in this community, and of course Richard discovered it with that first genome sequence, and Tim actually got the function validated, and brought in all of his skill set on that. But what you don't know is that when we first had these exciting findings, we came together at this American Phytopathological Society meeting in Austin, Texas in 2005, and we decided to get together and make a draft outline for that paper. So we went to a bar restaurant that I knew downtown Austin. We got an early start, I think it was about 11 o'clock in the morning. You guys were all jet lag. We were all jet lag. Tim was in the right time zone. But anyway, we went into this restaurant and took a table, and our server came up to our table, and the restaurant was almost empty. And the server came to our table and said, "Hey, look at that table over there. That's Laura Bush. Laura Bush is the wife of George W. Bush, the President of the United States. They're from Texas." And so there was Laura Bush sitting at a few tables over. And then after getting that message, we kind of looked around the restaurant again and saw there's like 3 or 4 big dudes. Each of them at a different table. All of them wearing dark sunglasses with big jackets on. Probably interesting hardware underneath. And now, by then we had 1 beer in us, and Richard, when he gets that first beer in, he gets pretty loquacious. And he started talking about his political feelings towards Mr. Bush, because it was about 1 year into that unfortunate war in Iraq that Mr. Bush started. And so Richard started talking in a much too loud voice about the idiot who's in the White House. And he's an idiot. I wonder what his wife is like. And Tim and I were just like, Secret Service, shut up, Richard. Like, he could not be shut down, but we did. my memory, please correct me if you have a different one, after Richard started his first harangue to the guys kind of straightened up a little bit, and in the restaurant there was mirrors everywhere. And if you looked at the 3D dudes and looked at the ways they were positioned in the tables, they could see into every mirror, into every corner of that restaurant. And I guess they were, probably some secret handshakes and signals were going on. But anyway, the good news is we did get Richard calmed down enough that we did not get arrested, because if he had gotten arrested, then he probably would have delayed that work from getting published. But finally got into drawing diagrams on napkins, what the figures would look like, some little outlines on other napkins, what the paper would look like, and a couple of weeks after that. So anyway, that's the lesser known story of Box. And then I just want to finish just quickly acknowledge the fact that also my work has depended upon the work of so many other people. Starting on the right side here are the people who I wrote letters to, or this is the day before emails. Anyway, so letters to, and actually they followed my instructions and made collections of isolates. 37 people around the world were able to set aside an hour or two of their valuable time and find cut to follow instructions. They collected of these and sent them to me in either Texas or Switzerland to isolate. And then I had 35 PhD students that had finished. Here they are in order of appearance in my lab. I've been really fortunate to have 17 different postdocs and over assistants, as we call them, here in Switzerland. I've been like a junior group leader, let's say. And some of you will be disappointed that you don't see your name show up over here in the close collaborators section. And that's because I only gave every name one position in this slide, so I didn't do double dipping. So even though I continued to work with Eva for many years since she was a PhD student, I don't just go over there and close collaborator and mission years. So please don't feel hurt if you don't see your name there. And I'm really pleased to say that among the 50-odd people who have been either postdocs or PhD students in our lab, about 13 of those people are now professors. And most of the remainder are lab leaders. They're PIs running either industry or government labs in different places around the world. And I could have done without all these people. So, thanks.
All right, well, I knew that was going to be about what I said. So we were supposed to have 40 minutes left for a discussion. We're 10 minutes late past the discussion. I will say that the majority of the stuff that was presented was factual, but I do have some alternative stories or alternative facts. But I can share those later since we're out of time. The poster session now has started 10 minutes ago. So go ahead and make your way over the posters. And thanks to the guys who presented.